Hi everyone. Today we're taking a closer look at one of the most important post-translational modifications, protein glycosylation, and how it reshapes the structure and function of proteins. Glycosylation refers to the enzymatic process by which monosaccharides or oligosaccharides are covalently attached to specific sites on biomolecules. In eukaryotic organisms, this modification occurs widely on proteins and lipids, playing essential roles in cell-to-cell recognition, signal transduction, and the regulation of molecular stability. Among the various forms of glycosylation, protein glycosylation is the most common and structurally complex. As a type of post-translational modification, it takes place in the endoplasmic reticulum and Golgi apparatus, after protein synthesis. By attaching diverse sugar chains to specific amino acid residues, glycosylation can profoundly alter a protein's three-dimensional conformation, biophysical properties, biological functions, and even its intracellular localization and stability. So, how does this modification actually occur? Depending on where the sugar chain is attached, protein glycosylation is generally classified into two major types, N-linked glycosylation and O-linked glycosylation. These two forms differ significantly in both their biosynthetic pathways and structural characteristics. N-linked glycosylation typically occurs at asparagine residues within a specific sequence. These sequences follow a defined pattern. Right next to the asparagine is an arbitrary amino acid except proline, commonly referred to as X, and right next to the arbitrary amino acid is a serine or threonine. O-linked glycosylation takes place at serine or threonine residues and does not require a defined sequence for recognition. N-linked glycosylation begins in the endoplasmic reticulum, where a pre-assembled oligosaccharide is transferred as a whole to a specific asparagine residue on the nascent protein, with the assistance of glycosyltransferases. The glycoprotein then enters the Golgi apparatus, where the glycan undergoes sequential trimming, elongation, and modification, ultimately forming hymanose, hybrid, or complex-type structures. O-linked glycosylation is typically initiated in the Golgi, where sugar residues are added one at a time to serine or threonine residues by specific glycosyltransferases. This process yields highly diverse and dynamically regulated glycan structures that vary significantly across cell types, developmental stages, and physiological conditions. Once fully processed, glycosylated proteins are directed to various cellular destinations, including the plasma membrane, lysosomes, secretory vesicles, or are secreted into the extracellular space. Glycosylation plays a pivotal role in modulating both the structural integrity and functional diversity of proteins. Structurally, glycans are bulky, hydrophilic molecules that attach to protein surfaces and significantly influence their conformation. They help stabilize specific conformations and reduce misfolding or aggregation. Glycans can also mask or expose key structural regions, altering how proteins interact with other molecules. For example, they may shield protease cleavage sites, increasing resistance to degradation, or help expose active sites for better recognition. Additionally, glycosylation improves thermal and pH stability, making proteins valuable in recombinant expression and drug development. Functionally, these structural modulations translate into diverse biological consequences. First, glycans modulate molecular recognition. Their highly specific structures influence whether, and how strongly, proteins bind to receptors, ligands, or antibodies. Second, glycans serve as postal codes, directing proteins to be secreted, anchored to membranes, or retained in organelles, like the endoplasmic reticulum. Many signaling receptors also rely on glycosylation to remain properly folded or membrane-bound. Without it, some fail to reach the cell surface or respond to external cues. In the immune system, glycans serve as critical identity markers, either facilitating self-recognition by immune cells or being subverted by pathogens and tumor cells for immune evasion. Aberrant glycosylation has been widely implicated in the pathogenesis of numerous diseases. The figure highlights glycosylation alterations observed across solid tumors, hematological malignancies, and autoimmune disorders. While glycosylation has drawn increasing scientific attention due to its biological significance, studying and characterizing it remains technically demanding. One major challenge lies in the structural complexity of glycans. A single glycoside can carry dozens of glycoforms, complicating structural interpretation. The lack of clear sequence motifs for glycosylation makes site prediction difficult and often leads to inefficient proteolytic digestion, hindering downstream analysis. In addition, glycoproteins and glycopeptides are typically present at low abundance. Enrichment techniques are often necessary to isolate them for detection. Even after enrichment, glycan analysis demands high-resolution mass spectrometry and specialized software for accurate data interpretation. Facing the complexity of glycosylation, MTOZ Biolabs provides a tailored, one-stop glycoproteomics solution. We identify various glycan types to address the diverse needs of basic research and disease mechanism studies. Our analytical platform integrates high-resolution mass spectrometers with nanoscale liquid chromatography systems for sensitive, high-throughput analysis. For upstream processing, we offer a wide range of enrichment and deglycosylation options to accommodate different sample types and experimental goals. In addition, our integrated bioinformatics support covers site-specific annotation, pathway analysis, and data visualization. From structural characterization to functional insight, we empower researchers to decode the biological significance of glycosylation with precision and confidence. In addition to glycosylation analysis, MTOZ Biolabs also offers other types of post-translational modification analysis, including phosphorylation and acetylation. Thank you for watching and feel free to contact us for more information.